What are these? You must have seen this type in the oceans where you have some shells that we were talking about yesterday, also a little bit about the shells. And you must have also come across hydra. Mostly we would have read about
Oceanic
hydra in our school.
creatures.
You must have seen hydra in inter also. Yeah, we have studied a lot about hydra. Why did we talk about hydra in inter and school times? Any idea? Any idea why we were talking a lot about the hydra in the school level? Anybody who can recollect? Did you have the subject about hydra in zoology? Did anybody read that? No idea?
Yes, ma'am.
Yeah. Why did we talk about hydra in our school days? No idea. We just talked about it as something like plant-like structures moving here and there, right? First thing is we talked about it because of its regeneration capacity. If you remember, you chop them into 100 parts, and each and every part again comes back to its life. So that is the importance of hydra over there when we talked about in inter days. Now here we are going to talk about its characters, general characters and classification, and they are in phylum Cnidarians. These are the shell-like structures, what you're seeing in the picture over here right now. Introduction over here, what are they? They are coelentrates, phylum Cnidaria, or they are also called as phylum coelentrates. Coelom is a cavity in between is called coela. Coel, C-O-E-L-E. Coel means cavity. Entrate means entry. So that is a gastric entry, or we were talking about some entry, hollow entries in porifera, spongocoels. So something like that. They have some entrance of structures of their seal is hollow. Hollowness in the center is called coelom. So coelentrate is, or it is also called as Cnidarians. Why Cnidarians? Cnidarians means something like needle-like structures which is able to sting us. They sting, right? So something like stinging capacity is called Cnidaria. N-I-D-A-R-A means needle-like stinging capacity is called Cnidaria, or it's also called as coelentrates because it is having a coelom or a hollow structure in between is called coelom. So that is coelentrates. They include diverse animals like jellyfish. You must have come across the jellyfishes, right?
Yes, Aurelia, ma'am.
Aurelia, yeah. Sea anemones, they are stationary ones. Corals, the most of the corals we come across, the red corals, the orange, the black corals, the beads that you come across, these are from the corals that we get. And most familiar is a hydrovia in phylum Cnidaria. And they are diploblastic. Diplo means two. So 2 layers, diploblastic eumetazoans with tissue grade of organization, because each and every tissue has some particular level of work that it has to do. So tissue grade of organization. And they are characterized by the presence of cnidocytes. Cnido, what I told is the cup-like structure, which is having a needle-like stinging capacity, which will sting to catch hold of any prey. So cnidocytes. The polyp and the medusa forms. Polyp is obelia. You must have come across obelia. I'll be showing that pictures, how they look like. So they have that polyp and medusa. Medusa is a cup or saucer-like structure is called as medusa forms. And you find almost 2,500 species of the coral split into 2 major groups. That is, some corals are hard ones and some are soft ones. So they are divided into 2 corals, that is hard corals and soft corals. And most of the corals can be found on the largest coral reef in the world, that is in Australia. It's also called as Australia's Great Barrier Reef. You must have come across this barrier reef over here. When you go over there and when you swim inside the ocean, you can see all this coral reef that you call, it's almost like a basement where you have thousands and thousands of coral reef embedded on them. And that's called as Australia's Greater Barrier Reef. Which is very famous when you talk about.
Yes, ma'am.
When you talk.
It's on east coast.
Right.
East coast of Australia.
Yeah. Okay. So what are the general characters? They're mostly marine and few are freshwater ones. Mostly colonial. So colony-like formation you can find on them. And sometimes they are single. or solitary corals. So this is a sea animal which is solitary, and this is the corals which are colonial in form. Diploblastic shows tissue grade organization, radially symmetrical. Now you should by now you should understand what is radial symmetry. When it is a
Two
round
equal
structure,
halves.
yeah,
Yeah.
radially you are going to cut it into two equal halves. But sea anemone shows biradial symmetry. Bi is something like this. You're going to cut it something like this, not like straight cutting. It's not a straight cut, but you're going to
It's
cut it
cross.
radial cross. So that's a biradial symmetry. And you can see different corals over here. This resembles like a brain of a human brain. So that is why it's called a brain coral. You find some branches over here called branch coral, tubular forms, tubular corals. And because it's very something like a sunflower, so sun corals and sea anemone. So these are some of the corals that you find under the sea. And
What is
mostly
meant by anemone?
anemone is just a name. There's a name
Just a
that
name.
resembles over there. Sea anemone. Yeah.
Okay.
Okay, so they are made up of polyp and medusa. This is a polyp form over here, and this is a medusa form. So in cnidarians, you have a polyp and a medusa form. Asexual form is the polyp form, and a sexual form is a medusa form over here. And polyps is a hydroid form, which is sessile. They are not moving, so they are stationary at one place, whereas the medusa is a moving type, motile one. And the mouth in the polyp is on the anterior side over here, and in a medusa, it is on the posterior side below. In the subarachnoid region, you have the mouth over there. And the body wall is made up of an outer epithelium, a gelatinous mesoglia in the center, and an inner gastrodermis layer is found over there inside. So you have an epidermis, an inner gastrodermis, and in between them is a mesoglia. We found this mesoglial layer also in the poriferans, and the mesoglial layer in poriferan was called as mesohalar region. Okay, so remember that mesohalar was the topic that we are talking about. Here we are going to talk about mesoglia. Why? Because it's going to be a jelly-like substance. So glia, mesoglial region. It's a thin structure in the polyps, and it is thick in the medusa form. The mesoglial region is very thick in the medusa form, whereas it is very thin. That light pink color you can see in between. So that is the mesoglial layer. So it's very thin in the polyp form and very thick in the medusa form. Why? We will be talking about that. Another important point is because of its thickness, it gives that buoyancy to the medusa so that they can swim in the water. They are not drowning inside. They are able to swim because of that thick medusa present in the medusa regions over there. So they help in buoyancy. Buoyancy means they can swim in the water. They don't drown inside. So that's the meaning of buoyancy over there. The body wall contains stinging cells called cnidocytes. So this is a polyp. These are the tentacles of the polyp, and each tentacle will be having the cnidocytes present. Now, if you take a closer look of this small piece over here, the edge, the ending region of the tentacles, you have that round stings-like structures over here. So when you take a closer look of this one part over here, they are made up of a capsule-like structure. And this capsule has a trigger-like structure, a sensory organ-like structure over here. And when this is triggered, when something touches them, they send signals to the coiled thread which is present inside the capsule. And once the signal goes and touches the thread is immediately stung out. So you can see this point has come out, it stings out, and it captures the prey. For example, if this is a prey, the flagella of that sting is going to capture it, winding around them, and then they discharge some poisonous substance into them, which makes the prey paralyzed. And once it has become paralyzed, when they're not moving, these are taken inside and they have it. So they act as catching the prey. So they help in catching the prey. So that is what I said. Cnidarians, if you can see the sting-like structures over here, cnidarians, something which will sting the body. So that is the reason why they're called cnidarians. And they help also in defense and capture of the prey. So they also help in defense mechanism, because any animal should have a defense mechanism, and also they should have something like how they are going to go and catch the prey. So all the animals should have something to defend themselves. Yeah, what is this lines over there that is coming out? Okay, the blind sac-like central cavity is called the coelenterons or the gastrovascular cavity. So this is a polyp form of the cnidarian. So this is the gastrovascular cavity, which is opening into a mouth. They have those tentacles around, and this is the red color ones in the gastrodermis. Whereas the outside one is the epidermis, and in between the purple one, light purple one, is the mesoglia that is present over there. And they also have some nerve network. So nerve network like structures found over there. And mouth serves as ingestion as well as ejections. So whether they are going to take in food or whether they're going to throw it out, everything is done in the same mouth. If you remember in porifera, we were talking only about the what is that, osculum, wherein
Yes.
only the ejection part is done, whereas the internal taking of the food is done only by ostia or the pores that are present on the body walls.
Osculum is for excreta only.
Ostia, I'm talking about ostia. Ostia is where
Acha.
the intercurrent canal is,
Internal.
water is coming inside, and
Yes,
osculum is where they are throwing them out.
yes,
But
excretion.
here the mouth is working as both ingestion and ejection of the unwanted waste materials. The medusa form coelentrates is specialized into stomach, so they have a proper stomach over here, the medusa forms, and they have the radial canals. You can see the radial canals coming this side and that side, the radial canal, and a round ring canal. Who is that, Ma? Can you please mute it? I don't know the name. Some Greek and Latin. IZN. Hello. Oh my God. Is it disturbing others? Others are getting disturbed? Yeah.
Yes, ma'am.
Who is this, Ma? There is no name on this. Can you please mute your
Device.
no answer? And it's okay. Hopefully. Yeah, she has said. Thank you. Okay, though, so they are made up of specialized stomach. Then they have the radial canals, the ring canal over there, and the ring canal. And this is the ring canal which goes around. Is somebody playing with their voice? Hello, who is that? IZNAVFP8N. What is that name? Is that any name? Can you please mute your system? Does anybody know that person's name? Your friends, anybody?
No, ma'am. We don't know.
Okay. Fine. Okay, so digestion. Yeah, the stomach helps the coelenterons. The coelenterons of there. Now you know what a coelentron is. What is a coelentron? It's a hollow structure that is present, helps in digestion and circulation. The digestion is the first extracellular in the coelenterons, and then intracellular is by the nutritive muscular cells of the gastrodermis. So in the coelentron, the first kind of digestion takes place. How? That is, when the tentacles gets in the food materials, they are pushed into the mouth, and from the mouth they go into the cavity over here, coelenterons, and then the gastrodermis that is present over here. This will help in digesting the food by the nutritive muscular cells. Exchange of respiration gases and elimination of the excretory waste occurs through body diffusion and the neurons. Now coming to the neurons over here, can you see the neurons over here? So this is the manubrium, or the entrance after the mouth. Then this is a mouth, then it goes into a manubrium and then into the stomach. And in this manubrium, you will find the nerve network on the manubrium and also on the umbrella shape over there, they have a lot of nerve networks which are interconnected between the epidermis and the gastrodermis or the endodermal canal. They have a nerve network which is connected in between by the mesoglia. The impulse conduction is a diffusion conduction. Diffusion is they are not connected by a single structure like that. So, for example, if you're talking about the human network nerve system, one nerve is going to give the information is conducted straight in a straight line. But here, the information is diffused. It's something like uh, the information is being distracted and they go around everywhere. So that is called as diffuse conduction. The nerve impulse can travel in any direction. When you're talking about the human brain, the nerve impulse cannot move here and there. It will go straight to the target. It will reach the target. It will not get confused. It will not get disturbed. Thankfully, our nerves, the axons, if you remember, are covered by a myelin sheet. Yes, you remember our axons are covered by a myelin sheet. That myelin sheet will not allow the information to be scattered here and there. For example, if our nervous system is something like this diffuse conduction, what will happen is, for example, if you are holding something hot iron rod, your brain has to give signals to your muscles on the hand to release so that you are safeguarding your skin. Okay. And the information comes directly to the muscles to your hands, and then you are you're throwing that rod away. Imagine your brain is giving information from here, it has to travel all the way to the muscles of here. If somebody disturbs you, if somebody is disturbing the information over there and that information goes somewhere else, you are still holding the iron hot rod. Imagine what is happening. You're burning your fingers. So thankfully, the human brain, the nervous system, the axons are myelinated so that the information goes straight to the target, reaches the target, and gives the information. They are not scattered. But here they have to scatter around so that they can move in the water as a peristatic movement. If you have seen a jellyfish, how they swim. So the information has to come all over the bell shapes so that they move here and there. So that is what is called as diffuse. Nerve impulse travel in any direction. Besides nerve networks, medusae have nerve rings and ganglia around the margin of the bell. So this is the margins of the bell over here. And the nerve networks in the medusa have ganglia around. So these are the ganglia. What do you mean by a ganglia? A bunch. Who's that? Who's that? Hello, Saida. Can you please mute your system? Saida Afra Ali.
No,
Yeah,
ma'am,
thank
it's
you.
not me.
Then who's that? From where is the sound coming?
I have muted myself.
Okay, then who's that? Why am I getting? Just ask somebody. Ask everyone to be careful when the class is going on, right?
Please, girls and boys, mute your devices.
Sheikh Mazhar, are you there? Or it is just switched on and they are doing some other work outside? Okay, because I see he has raised his hand in a form of a question. Anyways, okay. Where were we? Yeah. What do you mean by a ganglia? Ganglia is a bunch of neurons present in a globular forms. That's called a ganglia around the margin of the bells. So that is the meaning of ganglia over there. You must have seen in cockroach. We'll be talking about this in ganglia in cockroach and also in the prawns. There you will understand what exactly the ganglias are. Sensory structures like statocysts occur in the medusoid form. So this is a statocyst. What is a function of a statocyst over here? As a name indicates, they are sensory in nature. That is, anything touches them, they have a sense organs. Statocyst makes them. If the jellyfish is fallen upside down, what happens? They have to come back to its position. How do they come back to its position? It's because of the sensory structures like the statocyst, which tells the animal that they are upside down, come back to your position. Okay, something like what we have behind the brain, the cerebellum and the medulla oblongata behind the brain, which tells us our gait. If we are sitting, we are sitting. If we fall down, the brain tells that you have fallen down, get up and stand straight. That is the sensory action. So statocyst over here in the medusa works like that, which gives them a sense organ like structure. So if you can go through this picture over here, the outer side is the exumbellar, and that which is inside. So for example, if this is a cup-like structure, that is the medusa form, the outer one is a extraumbellar, and the inside form is a subumbellar region over there. And you have the tentacles over there. These are the tentacles. The tentacle bulb is present over there, the statocyst, the neurons in the tentacles. Then this is a circular ring formation or endodermal ring. And this is a velum. What do you mean by a velum or a velum? Velum is nothing but if you see in the Christian community during the marriage, they have that gungat like structures coming on top. So that is called the velum. So it has not much of work, but just a velum that is covering them. Yeah. So reproduction over here, they have both asexual and sexual reproduction over here. Budding, fragmentations, fissions, you find in asexual reproduction. That is budding, you can find in the polyp type, and sexual reproduction you find in the medusa type over here. Cnidarians are generally unisexual, that is either it's a male or a female, but sometimes you are going to see a bisexual type of cnidarians also. What do you mean by bisexual? They are hermaphrodites. Yesterday I was talking about hermaphrodites. If you remember, both the male and the female reproductive organs are present in the same animal. Fertilization external, that is the egg and sperm is going to fertilize outside, and the growth is seen outside the structure of the adult medusa over there. And development includes indirect and a free swimming ciliated larva called the planula larva. So the name of the larva over here, if you remember the poriferans, we were talking about 4 different larvas. Here you're going to find only one larva, that is the planula larva. So if they ask you to talk about the larval form of the cnidarians, you're going to talk about the planula larva. The species having polyp and medusa phases, alteration of asexually reproducing polyp. So this is a asexually reproducing polyp, and this is a sexually reproducing medusa, which is also called as metagenesis. And they have a remarkable power of regeneration. Now you know what exactly is a regeneration. Okay, coming to classification. So that was, we were talking about the general characters of cnidarians. Coelentrates, phylum coelentrates. Phylum coelentrates means that which is having hollow structure inside the body is called coelentrates. And why it is called as cnidarians? Because they have those stinging cnidocytes, which will sting the prey or also helps them in a defense mechanism. Now coming to classification, they are classified into 3 classes. As I said again, we are going to talk only about the up till the class. We are not going to talk about the orders. So the coelentrates are made up of, they are classified into 3 classes. First class is hydrozoa. Hydro means water animals. Hydro means water, zoa means animals, water animals. Examples over here are hydra, obelia, and physalia. Physalia, another name for Physalia is Portuguese man of war. Why Portuguese man of war? It resembles like that of a cap. If you had seen Pirates of Caribbean, the movie Pirates of Caribbean, there you will find all the pirates are having those cap, the long cap-like structures. So that is why they are called as Portuguese man of war. The second class is Scyphozoa. Aurelia is an example over there in Scyphozoa, and the third class is Anthozoas. Examples are Adamisia, or it's also called as sea anemone, and corals. So we'll talk about one in a very short gist. Hydrozoa, as I told you, water animals, mostly marine. Some are living fresh in fresh waters, chiefly colonial. They form in colony formations, and some appear solitary. Medusa stage is absent in few animals. Sometimes both polyp and medusa are present in some. So both are there. This is there, that is there. A little bit of this and a little bit of that. Okay. Mesoglea is acellular. Now you know what a mesoglea is. The space between the epidermis and the endodermis is a mesoderm. That's called as a mesoglea. This is acellular. Acellular means they are made up of only single type of cell. That is, they are made up of amoebocytes. Remember the amoebocytes in the porifera, where they help in digestion of the food, where the conocytes, with the help of the flagella, pulls the food inside the mesoglial stage, where the amoeboid does the nutrition part of it, digestion part, and sending the nutrition to the whole part of the body. So in the same way, mesoglea over here is acellular, having the amoebocytes, and cnidocytes are restricted to the epidermis. So the cnidocytes are only on the outer covering of the cnidarians. Why? Because they have to be there to help in protecting themselves and also to capture the food. So that is the reason why they are only on the epidermal layer. Gonads also occur in the epidermal layer because fertilization is external. So gonads are also on the epidermal layer. The colonies are polymorphic with different types of zooids. Polymorphic. Poly means many types, with different types of zooids. Zooids are something like they are having some different tissue organization. Each tissue is made up in such a way they have some specific work that has to be done. So that are called as zooids over here. Like gastrozoids. Now these gastrozoids are one gastro means food. So feeding type of gastrozoids. Dactylo means defensive type, and gonads, reproductive type of zooids. So they are having these three important zooids in any given cnidarians over there. And to maintain the equilibrium, they possess statosis. Now you know why I told maintain equilibrium. Equilibrium means when they are swimming in the water, they should know in which position they are, whether they are upright, whether they are sideways, or whether they are upside down, topsy-turvy. So they help in bringing the animal back to the position. So that is called statocyst. So you can make an underline and make a note of it in your rough book. Statocyst. Okay. Examples over here: Hydra, Obelia, Physalia, and Tubularia are the examples over here, how they look like. So this is an Obelia, where you have the tentacles over there, then you have the polyps present over there, the perisacral annuli, then you have the medusa buds, the budding coming out, you have the gonophore, the blastocyle. So this is an Obelia over here, whereas you have a Hydra over here, different type of Hydra. And then this is what I was talking about, the Physalia or the Portuguese man of war. Can you see the cap-like structure that is seen over there? So it resembles like a cap, the war cap. So that is why it's called as the Portuguese man of war. And what you find over here, they have a pneumatophore. That is, there is a space over there inside. It is just a balloon-like structure where air is filled. More than a balloon, you have those hot air balloon which flies on top, right? And the hot air goes inside, which makes them open up, and then they are able to fly in the air. In the same way, these Physalias have pneumatophores. That is, there is a gap inside. If you can see a transparent Physalia over here, inside it is filled with gas that helps in buoyancy in the water, so that they are swimming in the water. Okay. They have the small dactylozoids. Now tell me, what is the function of a dactylozoids? I was talking about gastrozoids. Gastrozoids are feeding in nature. Gonozoids for reproduction. What is the help of a gastrozoid? Dactylozoids? Anybody? They are helping in, they act as sensory organs. They help in locating and capturing the prey. Prey is not preying. A prey is something, a small animal, which a larger animal will catch hold of them and eat them. For example, when you're talking about a lion and a deer, a lion is going to eat the prey. Prey is the deer. So they're going to eat the prey. Right? So hunting, fine. But when I'm talking about what a prey is, for example, if you see catching prey, P-R-E-Y, it's not P-R-A-Y, it is P-R-E-Y. Prey is the one that is being eaten away. So that's the prey. So here, when there is a lion and a deer is a prey for the lion. They're going to eat. Fine. So that is what it is all about. Okay. Now, if you can see that this Physalia has some more tentacles over here other than the dactylozoids, the gastrozoids, the long ones are the gastrozoids, the small tentacles are called the dactylozoids, and the larger ones are the gonozoids through which the sperms and the ova, the egg is released. And then other than that, they also have this larger dactylozoids. This larger dactylozoids helps in holding the prey if it is trying to escape from its mouth. They have those big tentacles which goes and capture them. So that is a meaning why they have this larger dactylozoids. And this tentacles, some tentacles are bearing battery-like structures called nematocysts. So you can see those battery-like structures over here, which is called as nematocysts. What is the help of a nematocyst over here? Nematocyst is nothing but nidariocytes, or the stinging cysts, what you call them, stinging cells. So nematocyst or nidocyst is one and the same. I hope it is understandable for you all. Did you all understand this point? All the zooids? Any doubt in the zooids? Fine. If anybody is having any doubts, please ask me to stop and ask your question so that I can explain it to you all again. Don't fear me. So I'm here to help you all. Okay, going to the next class, the class Scyphozoa. All the animals belonging to this class are marine in nature, and Medusa stage is predominant in this particular class. Medusa stage is predominant, and mouth is surrounded by 4 oral arms. If you can see over here, they have this oral arms. So these are called the oral arms over here. So this transparent one over here, these are the oral arms. Mesoglia is cellular and contains amoebocytes, and this should be acellular, not cellular. There is a print mistake over here, acellular. It is going to contain only amoebocytes. Nidocytes occur in the epidermis and also in the gastrodermis. Nidocytes, I hope now you know what a nidocyte is. The stinging cells is called a nidocyte, which occurs both in the epidermis and in the gastrodermis. Why do you need a nidocyte in a gastrodermis? Because if there is any food particle that has entered the stomach directly, then the nidocytes which are present in the gastrodermal layer inside the stomach walls, they are going to sting them, paralyze them, and they're going to eat them. So that is the reason why they have the gastrodermis over there. Gonads occur in the gastrodermal region. So you have the gonads present in the gastrodermal region, and to maintain equilibrium, they also have tentaculocytes. What was the equilibrium cells that you found in the hydrozoa? Anybody? What is the name?
Statocyst, ma'am.
Very good, statocyst. Yeah. So in hydrozoa, you have a statocyst, and the same name over here in Scyphozoa, it is called as tentaculocyst. So you can just make a note of that. To maintain the equilibrium, you have a tentaculocyst in the Scyphozoa. Polyps are solitary or may also exist in colonies. Examples are the jellyfish over here, the Aurelia, and the Rhizostoma. So we are going to talk a little bit more about type study of Aurelia over here. So this Aurelia is very important for us. You have a jellyfish and Rhizostoma. Do you know some important facts about jellyfish? Most of the swimmers in the ocean who go out there for sunbath and who go there for enjoying and all, when they're going into a vacation, most of the people are stung by this jellyfish. So severe is the stinging, what is that stinging one? The dactylozoids. So there are such severe stings. Sometimes it's a simple wound. Sometimes it can be little bit serious. Sometimes a person can even die of the sting. So you have to be very careful with a jellyfish. Never think that they are so cute, so lovely, umbrella shape. You don't have to go and touch them because they're going to sting us and kill us. So you have to be very careful with this jellyfishes. Most of the death in the ocean occurs because of the stinging of this jellyfish. And you have this Aurelia and Rhizostoma examples. Coming to Anthozoa. So what are these anthozoans over there? They are marine, solitary, and colonial. I have also given what is the meaning of all these hydrozoa, Scyphozoa, and anthozoa. Okay, and they are sedentary, polyploid forms. Medusa stage is absent, so they don't have Medusa stage over here. Mouth is oval and is surrounded by a whirl of tentacles. Can you see those whirl of tentacles? So this resembles like that of a flower. So that is the reason why it's called anthozoa. Antho, flower-like animals is anthozoa over there. And this petal-like structures is nothing but the tentacles. That is the formation of tentacles over there. And hence the name anthozoa. Mouth leads into the tubular pharynx called stomodium, that in turn opens into a coelenteron, hollow. Okay, so that's called coelenteron. Divided into radial compartments by vertical septa called the mesentery. So they have those radial compartments separated by mesenteries. Cnidocytes occur in epidermal wall as well as gastrodermal regions. Gonads occur in the gastrodermis. And the examples are monactus, this is a sea anemone, telesto, so this is a telesto over here, and tubipora, tube-like structures, so tubipora, and xenia over here. So these are the examples of anthozoa. And coming to, so that was the characters, general characters and classification. You have three classification: hydrozoa, anthozoa, and scyphozoa. So you're going to talk about it. Now, when you're talking about the type study, we are going to talk about one of the scyphozoa type study over here. So under scyphozoa, we are going to talk about Aurelia, kingdom Animalia. Why Animalia? Because we are going to talk about animals. So kingdom Animalia, phylum Nidaria, because all these are going to have those nidocytes, so the stinging rays. So they come under Nidaria. Class Scyphozoa, and order Simistomae, family Ulmeridae, genus Aurelia, species Aurita, Aurelia, Aurita. Always species has to be written in a small letter and not a capital letter. So whenever you're going to write the classification, remember genus in the capital letter starts with a capital letter, species with a small letter. So that is the reason why they have written that A as small. Aurita, Aurelium, Aurita is a example that you're going to talk about in type study of Aurelia. So this is a lovely umbrella shaped transparent structure that is flying, that is swimming in the water. So all these small dots are nothing but the Aurelia that is present in the ocean. Lovely to see them in the ocean. And habit and habitat. Common jellyfish. So what is a common name of Aurelia? It's common jellyfish, or also moon jelly, because they resemble like that of a moon. So it's called as moon jelly. Why is this lines present on my slides? Can anybody see the lines on the slides?
Yes, ma'am,
Yes,
it's
ma'am.
plenty.
Someone is playing with your.
I know. Who's that? Is anybody, any child sitting over there who's playing with my slides? I don't need any cracks on my slides, okay? Please, whoever's doing that, try to rub that up. I don't know. Please don't keep your children in front of the camera and you people working over there. The class is for y'all, not for your kids over there. Whoever's doing it, please rub it. Okay, and where was I? Yeah, moon jelly. So they resemble like that of a moon over here. So that's why it's called as a moon jelly. And they are present in the tropical and temperate oceans of the world, close to the surface of the water. So that is why you can see they are swimming in front of the water over there, right? So it is a cosmopolitan in distribution. You can find them everywhere, lives either singly or in large groups, floating or swimming freely. They are carnivorous. They try to eat any animals, small animals that come in their way. Reproduction, both asexual and sexual reproduction. Shape, they are flattened, bowl or saucer shaped, gelatinous structure. Gelatin means sticky structures, which is also called as bell or umbrella shape. Exhibits tetramerous radial symmetry. So if you can see that radial symmetry over there, this is the radial. So tetramerous, tetra is 4, tetramerous radial symmetry. And it is usually about 7.5 to 10 centimeters in diameter. So diameter is up to 7.5 to 10 centimeters, though much larger forms are present, but these are mostly found in the water. And recorded in Atlantic coast, almost transparent, having bluish white body with pinkish gonads. So that's how they are being differentiated. Okay, external structure over here, distinctly divided into a slightly convex. So what is this? This is a convex structure. So when I'm talking about a cup like this, the convex structure is an ex umbrella stage, and a concave, which is present inside the concave, is called as a sub umbrella surface. So ex umbrella and sub umbrella surface is how you are going to name them. The margin of the umbrella is circular, which is broken by 8 notches. So you have some notches like structures over here. 8 notches are present around the whole of this aurelia over there. And each notch is provided by a pair of marginal lappets. They have a notch, and the notch is covered by a flap like this, lappets. So that is what it's called as, lappets, enclosing the sense organ called tentaculosis or rophalium. So either it is called as statocyst, or it is called as a tentaculosis, or it's also called as rophalium. So make a note of this, rophalium. The margin of the umbrella in between the notches provided with numerous small closely arranged marginal tentacles. Now you can see the small tentacles like structures. So these are the marginal tentacles, and the margin of the umbrella is bearing marginal lappets. Lappets, in between the lappets you have those tentaculosis. Okay. And then a thin ridge like velarium or pseudo velarium. So this is a small thin structure of velarium or pseudo velarium. Again, I'm telling you, a velarium is a cover like structure. Anika Zaki is there, and they are making some lines on my screen. Anika Zaki, who's that? Please try to remove all those lines on my screen. Can I remove these lines? Is it possible?
Yes, ma'am, it's possible.
Yes, ma'am.
Can you all also do it?
No,
No,
ma'am.
ma'am.
We don't have access.
Okay, how do I remove that? Is that by any rubber or something like
Ma'am,
that?
go in the view options.
No, ma'am. It's a picture of pencil.
Huh.
Can you touch it? If you touch it,
We should go in view options and annotate. In that
Huh,
you'll get eraser.
yes.
Yeah. I have a rubber over there. Yeah. So till then, just go through the pictures over there as I'm cleaning my whiteboard over there. Okay. Fine. Yeah. Now my board is clean. Okay. This is how the Aurelia Munjali looks like over here. The epidermis, the outer epidermis, in the middle you have the mesoglea, and then the inside is a gastrodermis. You have a gastric cavity that is present, that is called a coelentron, or the gastric cavity. The outer one is a exumbellar. Then you have a radial canal, which is the radial like structure, something like that. So you have a radial canal over there, and the whole one is surrounded by a circular canal. So this is a circular canal. And you have those gonads. 4 gonads are found on either side of the mouth. So 4 gonads over there. You have those tentacles, the mouth. The mouth is covered by oral arm like this. So you have 4 oral arms. This side of the oral arm is chopped off so that you can see what is there inside. And this is a tentaculocyst, or it's also called as rophalium or rophalium over there, tentaculocyst, which is sensory in nature. Now, if you see the subumbrella region, if I'm going to show you the inside of the aurelium over here, it is going to look something like that. You have a mouth over there, the central mouth, which leads into the gastric cavity inside. The mouth has 4 corners. 4 corners are having some radial canals over there. Then, yeah, so this is the radial canal that is having. And the mouth corners will be having per-radial canals. I'll show you how the per-radial canals looks like. Then there are gonads, 4 gonads, which is in the shape of a horseshoe shape. So that's the 4 gonads, which is pink in color if you see a live aurelium over there. And this is the rophalium. So you have on all either sides the 8 tentaculocysts or the rophalium that you find around, and the small ones are the tentacles, and these are covered by pseudovelum. And these are the ad radials. These are all, what do you mean by these radial canals is from where the water will enter into the whole body parts of the aurelia, because 96% of the body is covered with water, the aurelium. Hence, because of these canal systems that is present all over the body of the aurelium, the whole body is filled with water. And this is provided with the help of this canal systems. So what are the different types of canal system? They have the radial canals, they have the perradial canals coming from this side. I'll show you all those pictures over there. And between the radial and the perradial, you have small channels like structures over here. That's called an ad radials. So this is the picture of how an aurelia looks like. So manubrium, the subumbrella surface over here. So this is the subumbrella surface. The mouth is nothing but the manubrium over there. The center or subumbrella surface having a well-developed, short, inconspicuous manubrium bearing a large squarish mouth. So this is a square-shaped mouth present over there. Oral arms, you can see the oral arms on all the 4 sides of the mouth. You have the arms extending. So that's an oral arm over there. And they are frilled and tapering process. And they are also provided with numerous nematocysts over there. Gonads and subgenital pits over there, the horseshoe shape, that's what I was talking about. This is the gonads, which is having a horseshoe shaped gonads present over there. And they are provided with thread-like gastric filaments bearing nematocysts in the gastric pouches. Now, coming to the canals over here, how does a canal look like? If you can see over here, they have those long channels over here. So this is the square-shaped mouth, and the mouth is covered on all the 4 sides with gonads over there. And on the straight structures over here, this is the single canal over here from this point to this point, and from this point to this point. All these 4 canals are called interradial canals. This canal, this canal, this canal, and this canal are the interradial canals. And opposite to this canals, that is on the corners of the mouth, you can see one more canals coming outside on the corners of the mouth. So this is the corner mouth. So this canals are called as perradial canals. And between this adradials and the perradials, you have this in between the radials and the perradials, you have this adradial canal. So this is a interradial canal. This is this is a perradial canals, and in between you have this adradial canals. So these are the different canals which will help in getting the water inside and allowing all the water to be surrounded by whole part of the body. So you have to remember what is an adradial, what is an interradial, and a perradial. Interradial is a cross, complete cross. Perradial is coming as a cross, that is from the corners of the mouth. You have those radials canals coming out, so they are called as radial canal, and a straight line cross is called as interradial. And in between these two, whatever the lines are coming is an adradial canal. I hope you have understood that one. Okay, so coming to histology, what do you mean by histology? I am going to talk about internal structure of the aurelium. Till now we saw the anatomy part. Now I'm going to take a section of it. That is, for example, if this is aurelia, I'm going to cut them and see what is there inside, how the internal structure looks like. So that is the histology over there. So you have an ectoderm that covers the bell all around. So this is the ex-umbellar surface, and that covering them is the ectoderm. In between you have a mesoderm. So as we talked about in the characters, we saw that the mesoglial structure is thicker in the aurelium or a medusa form over there. So this is the thick, the black color ones is a mesoglia, the thick mesoglia over there. And after that mesoglia, you have a endoderm inside. The covering part of the body is the endoderm. Then this is the central stomach, which is present over there, and it has a gastric filament which is filled with that nematocyst. And this is a gastric pouch. The gastric pouch is where all the digested food is stored in a form of fat and starch. And they have this gonads, the gonads that we saw in the horseshoe shaped, that's a gonad inside. Then this is an interradial, the cross is an interradial. Then you have the circular canal, the whole umbrella is having a circular canal because it has to be interconnected. So you have a circular canal. And you have a hood over there, which is having, that is the lappets, which encloses the tentaculosis in between. So that's a hood over there. And inside is a subumbellar surface, opens into a mouth, because in the polyps you have mouth on the upper region, whereas in the medusa form the mouth is on the posterior region. So the mouth leads into the gullet, and this is a subgenital pit. So this is about the histology of the medusa over there. You can just go through that, because whatever I told is what is present over there. The endoderm, which is a columnar ciliated epithelial cells, and they are made up of cavities of tentaculosis and confined only to the gastric filaments or inside the stomach. Mesoglia, filled in between the ectoderm and endoderm, has the amoebocytes. What is the function of an amoebocyte? It helps in digestion, capturing the food and digesting the food, and transporting the food and waste material out of the water, out of the medusa. Nematocysts in Aurelia are found distributed in the oral arms, X and subumbellar surfaces, on the marginal tentacles, tentaculosis, and also on the gastric filaments. So these nematocysts are made up of 3 types. Don't get confused with the tentaculosis and the nematocyst. Tentaculosis, it is sensory in nature. When you're talking about nematocyst, we are talking about nidocyst, needle-like structure. So nematocyst and nidocyst are one and the same. The name differs according to the way they are explaining. So nematocyst, stinging cells. There are 3 types of here. Atrichus without spines is called atrichus. That is the flagella which comes out of the capsule. The flagella doesn't have any spines in them, so that is called as atrichus isorhiza. Elongated capsule, and there is no butt-like structure which helps in holding the flagella on them. So they don't have a butt, and the thread is without spines, and it opens at the tip. So this picture over here, what you can see over here, is a atrichus capsule. So this is a oval shaped capsule with a long thread-like tube, opens outside over there, and they don't have spines on them. So that is the reason why they are called as atrichus. A means absence of the spines. Now coming to heterotrichus, hetero. So this is a heterotrichus, a small capsule and a small butt is present over there. So this is a small butt over there, and a long spiny thread-like structure comes out, and they have those spines. Irregular spines are present in the heterotrichus. So that is the reason why they are called hetero. Hetero means many types. So the spines are not of the same shape and size, so that that is the reason why they are called as heterotrichus type of isorhiza. And the next one is a holotrichus isorhiza. What's a holo? So a capsule over there, a long butt, and then they have those long thread-like structures. The spines are all same structure, same size, so that is why they are called as holotrichus. And also they have a larger butt, which is also again narrow and large. So that is the reason why they are called oval capsules or holotrichus isorhiza. So these are the 3 types of nematocysts that is present in the medusa. Coming to gastrovascular canal, gastrovascular. Gastro means stomach, vascular means the movement, the vascular system, or the blood vascular system, what we call, where the blood has to move from one place to another. Vascular system, the vessels, the blood vessels over here. So here the vessels are nothing but the radial canals, the interradial, superradial, and adradial. So that is why you call as gastrovascular canal. Mouth leads into a small gullet within the manubrium. Manubrium is a small opening before the mouth, so lip-like structures. So manubrium over there, finally opens into a spacious stomach, and the stomach occupies a greater central part of the aurelia over there, and they have those four oral structures, oral arm that covers the mouth over there, and at the four corners of the mouth they have perradials and interradials. I've talked about all those, what are they. So they have those radial systems over there. You can see the picture over there. And then these 8 canals are much branched and they end. What are the 8 canals? The adradial canals. They are branched. Each 8 unbranched adradial canal arise, 2 from each gastric pouch. That is, if they have a gastric pouch, 2 from here, 2 from there, 2 from there. So all the 4 side corners, 2, 2. So they end up having 8 unbranched between the interradial and the perradial canals. And all these 16 canals, what are the 16 canals? 8 of the adradials and 8 of the perradial and the interradial canals. So they all sum up to 16 canals. They open circular or in a ring canal situated at the margin of the umbrella. So this is the ring canal. So all the adradials and the perradials, can you see, they are opening into the ring canal. So they're all connected. So this is the ring canal. And the gullet, the stomach with gastric pouches and all the canals constitutes the gastrovascular canal, and the entron cavity except the gullet is lined with ciliated endodermal cells, so that it pulls the water current inside them. Musculature of the body is well developed, confined to subumbellar surface only, not outside but only inside. Subumbellar surface consists of striated and non-striated muscles. Striated and non-striated, if you talk about the striations of the muscles, you have the proteins called the myosin and the actin proteins, which gives us that striation like structures, line structures over there. So they are made up of either striated or non-striated. That is no striations in the muscles. Circulation, how does a circulation occur? So with the help of the beating of the cilia in the endodermal lining of the gastric channel over there, what happens is water is drawn into the mouth. So first water is drawn into the mouth, and into the mouth they go pass into the gullet, and into the gullet they go into the stomach over there. From the stomach they enter the gastric pouch, a small gastric pouch. Then they enter into the adradials. So these are the adradials. They are moving into adradials. From the adradials, they go into the circular canals. From the circular canals, they are going into the interradials and the perradials, and then into the gullet. They go into the gullet, and from the gullet, they are excellent grooves in the oral arms that you find over there. And in that the water takes in the food and removes the water waste substance again from the gullet outside. So they enter into the gullet, and again they go out of the gullet from the interradial and the perradial gullet and outside. When they are coming inside into the stomach, and then they go into the adradials, the small 8 adradials. When the water is going into the adradials, it brings along with that the food materials, the nutrition is taken in, diffusion occurs, oxygen is taken in, and then as they are moving outside, they go into the perradial and the interradial to the gullet and then out of the body. So that is what is circulation in aurelia is all about. Nutrition, as the food is being brought in, that is when the water comes in and also the tentacles capture the food and brings it to the mouth, the food is taken in. Carnivorous is what we were talking about as the characters. What are they? Eggs, fish, small larvae, and pieces of animals. Here when you're talking about pieces of animal, it can also be a chitin. Remember a chitin? The hard covering that you find on the cockroach? Yes, that wings that is present on the cockroach. Yeah, so that's a chitin. They're able to even digest that particular chitin. They say even if the world comes to an end, chitin cannot be destroyed. Yes, Arif, you have any question?
Ma'am, are there 4 adradials or 8?
8 adradials.
8. Okay,
8.
thank you.
Yeah. and 4 perradials and 4 interradials. So that makes up to 16 radial canals. Got it?
Yes, ma'am.
Okay. The food is caught by the oral arms and tentacles and transferred to the mouth. So oral arms, that is in the mouth, they have those 4 oral arms. The oral arms also can capture the prey, and also the long tentacles can also capture the prey and bring it to the mouth. Ciliary feeding also takes place, and ciliary how? When the animal is moving, any planktons, the phytoplanktons. What are the phytoplanktons? It can be an algae, flagella, the small phytoplanktons that you find in the water, which makes the water green in color, right? Because of that algae present. They get stuck up. Once they get stuck up to the body, what happens is the cilia moves them into the mouth, and then it is taken inside. So different variations. As I told you all in the first class, why are animals able to move from one place to another is because in search of food. They have to survive. For that, they have to keep moving. So when they are moving, there are some planktons that gets trapped onto the surface of the body, and with the help of the mucus that is being secreted, they are able to push them into the mouth with the help of the cilia, and then they take in. So food is taken inside the mouth. And the gastric filaments kills or paralyzes the living organisms, and they are devoured or they are taken inside. Endoderm of gastric filaments and enteron produces digestive juices. So digestive juices can be hydrochloric acid. It depends, whatever the gases that they have, the juices that they have, they secrete them, and they dissolve the food and they eat the food. Partly digested food has been broken down into small filaments and swept by the cilia into the canals, and the food particles are ingested into the food vacuoles by endodermal cells. Endoderm, inside the layer, inside the mesoglia, you have an endoderm. So your food vacuoles are present over there, and the digestion occurs in that way. Enzymes digest the proteins, carbohydrates, and fats, and even chitin. That's what I was talking about. The chitin, the covering of the insects is called as a chitin. And the digested food is taken by the amoebocytes. Where do you find the amoebocytes? In the mesoglial regions, and distributed accordingly to the whole part of the body. Endoderm of the gastric pouches stores reserve food as fat, droplets, and glycogen. The same thing that you find in higher vertebrates also, glycogen. And the gastrovascular system fulfills the function of digestion and circulation. So what is happening to the respiration and excretion? Respiration and excretion occurs when the water is entering inside the body. As the water is entering inside, the water also carries oxygen during the circulation, and it also removes the carbon dioxide. The oxygen requirement in medusa is very less, as I told you, because the whole part of the body is made up of 96% of the water, and water means definitely oxygen is dissolved in them. So it needs only limited amount of oxygen when it is taking in the water. And the gastrovascular system also removes the excretory matter. Coming to the nervous system, very simple nervous system over here. You will find some nerve networks in between over here, the nerve networks, and in between the these tentaculocytes, the rofallium, in between that very thin transparent one, you can see that's a nerve network, also present in the circular muscles on the subumbellar region. So they are present in the subumbellar region. The nerve networks comes from the ectoderm and the endoderm, and they form a network in the mesoglial region. In between the mesoglial region, they form a network, so that the information is passed on as a diffused information. So the whole part, if one part gets some information, the information is distributed wholly. Diffusion occurs over there. It is not information over there and the sensory action is just over there. It's not like that. It is completely giving. The whole medusa is going to give that sensory action. So diffusion. That is the reason where they have this nerve network-like structure where everything is interconnected. And it controls the local reactions and also feeding. So whenever they come in contact with some sensory actions, they tell them that there is something present over here, and they are able to catch the prey. So feeding also is helpful with this nervous system. Sense organs. So here we are going to talk a little bit about tentaculocytes, or it's also called as rofallia. Tentaculocytes, or also called as statocysts in hydrozoa. Characteristic sense organs. Yes, ma, what's the question? Somebody raise a question? No? Okay. The Raphalia is a characteristic sense organs of the Scyphozoa. So yeah, the Raphalia is present somewhere over here. The statocyst, or the Raphalia, or the tentaculosis is a club shaped over here. So something over here in the marginal lappets, you have those tentaculosis present. How many tentaculosis I told? There are about 8 tentaculosis around the circular region of the aurelia over there. And each lying between paired marginal lappets. So this is a marginal lappets. Marginal lappets is, for example, can you see this lappet over here? So this is the lappet that I was talking about, and the tentaculosis is present over here. So this is a lappet, like something like that, covers the tentaculosis. Okay. And at the ends of interradial and perradial canal, at the ends of interradial and perradial canal, you have those 8 tentaculocytes, and all these tentaculocytes are covered by the marginal lappets. They are small modified tentacles. The tentaculocytes are also called as small modified tentacles. That's why the name tentaculocytes. The edge of the bell projects over the tentaculocytes as a hood. So the tentaculocytes is covering as a hood, and several sense organs are localized near the tentaculocytes or the sense organs. They're very, very sensitive. That's why they are sensory in action. And the small granules you find, so those granules are called as statoliths. Now, statoliths is something like our inner eardrum, the eardrum, where it tells us whether we are sitting upright or we are slanting or we have fallen down. So that is the one that tells us the equilibrium is maintained. For example, if somebody is going to punch you hard into the ear, you will have a kind of convulsions and chakkar jaisa, you just fall down, right? That is, you have lost your center of gravity and then you fall down. Then after falling down, the statoliths of your, in case of Aurelia, or in case of us, we have the eardrum over there, which tells us that you have fallen down, get up and stand straight. So this is helpful over here. The statoliths in the Aurelia helps them into getting back to their position. So here you can see there are 2 olfactory pits over there. So they have this olfactory pits, extended olfactory pits over here. So here what I'm showing you is a tentaculocyst or Raphalia. There you can find there's a radial canal coming over here. As they said, tentaculosis is present only in the radial canals, that is the interradials and the perradial canals. And they have an outer sensory pit, and they have a ocellus, a pigment called ocellus over there. And this is the Raphael lappets. And inside you have those tentaculocyst, which is present. So the whole structure over here is one tentaculocyst. So this is what he has told over there. The pigment spot or ocellus. So this is a pigment spot, or also called as ocellus, which is a photoreceptor cell, where the light, whenever it needs some light, the photoreceptor cells, they will say that the light is coming over here, so you have to go and swim over there. So sensory in action for photoreceptors, which is also called as sensitive to light. And there is this tentaculocyst is made up of 2 parts, most important parts: a club shaped lithostyle. So a club, can you see this club shape here? You can see this club shape over here like this, but here you can clearly see the club shaped lithostyle or statocyst, containing an endodermal, inside an endodermal calcareous otolith or pigment cup. So this is a black color one, otolith or pigment cup. What it does over here is they act as a sensory in nature, which is made up of sensory pad. So what happens is, when they are moving downwards like that, for example, if you see over there, so this is a statocyst over here. When the animal is in straight position, the tentaculosis is in straight position, but when it is falling down like this, the sensory nerves touches the tentaculosis. They get a signal over there saying that the body is moved upside down. They are going to come back straight. So that is what is called as sensory in action. So that is all about this tentaculosis with a lithostyle or statosis, is also called as statosis, and which is having an endodermal otolith. The statosis, you will also come across the same statosis in prawns. When you're talking about the arthropods, when I'm talking about the prawns, we'll come across this. Then again, I'll ask you all what is the function of a statosis. It is sensory in action. So what happens over here? When the animal is tilted, the lithostyle presses against the sensory pad, and when the two together work as an organ of equilibrium, with the result that the bell automatically turns itself upright. So that is the tentaculocytes. Coming to reproduction of the Aurelia, type study Aurelia reproduction. When you're talking about the reproduction, am I running out of time? You want me to continue, or shall I continue for 10 more minutes?
Yes, ma'am, you can continue and
Yeah,
just complete the topic.
Yeah, we'll complete the topic so that we will be ready with the next topic in the next class. Okay, so
Yes.
please bear up with me. You people don't have to go anywhere, right? So let me complete this. Sunday bahar jane ka thode din ke liye rok do, so that we will complete our chapters. Okay, coming to reproduction. When you're talking about the reproduction, here this picture is an example of sexual reproduction, where you can find two adult Aurelia present over here. And as we said, they are unisexual. So the male Aurelia is going to give out the sperms, and the sperms enter inside the female Aurelia and fertilizes the ova over there. Once the fertilization is occurred, it comes out through the gastric pouch, from there into the mouth, and from the mouth they are thrown outside, and the eggs are coming outside. And once the egg comes outside, the zygote divides its cells from morula stage to blastula stage. The bundle of balls is called the blastula stage. And from the morula to blastula, it enters into gastrulation stage. What is a gastrulation? Invagination of the cells. So when they invaginate, it's called as a gastrulation stage. When they invaginate is when you have those three germ layers forming, that is the ecto, meso, and the endoderm. And after the divisions, you have a free swimming ciliated planula larva over here. So ciliated, you can find all those cilia that is present on the body, ciliated planula larva. And once the planula larva has been grown, they go and attach to a substratum. So this planula larva goes and attaches to the substratum over here, and once they get attached, the cells again divide itself to form the next stage, what you call is scyphistoma. Scyphistoma resembles like a polyp stage, where it resembles like a vase-like structure over there with the help of tentacles. They have too many tentacles around, which helps in capturing the food, and digestion occurs over there for the help of the nutrition. And as they are growing, they change slowly into strobila stage. That is, the segments are being formed. This scyphistoma starts to bud, and as they are budding, they form this segment-like structures over there. And this is called as a young strobila stage. And this segments again increases to form as ephyra budding over here, ephyra budding. That is, strobilation occurs over here. From scyphistoma to development of ephyra budding is called as strobilization. And this strobilation is where you have those different buds. And as the adult has grown from a young strobila to an adult strobila, each ephyra separates from the adult strobila, and they are released in the water. So they become free ephyra. See, this each segment is one ephyra. So all these buds will be actually, it is attached by some muscular strings, and once they are ready to be chopped off from the parent strobila, what happens is the muscular strings are released and they are flying out. So each one, something like when you're removing each petal from a sunflower, so you're going to remove like that. So in the same way, each budding, each layer is one ephyra. and they are removed out by budding, and they are freely swimming in the water, and after some time, after some development that is going on in the ephyra, they change into an adult male or a female Aurelia. So this is the sexual reproduction. So coming again, the sperms fertilizing the ova. Once the ova zygote is formed, the zygote swims out through the mouth of the adult female Aurelia, and once into the water, they start dividing into two cell, full cell, morula, blastula, blastocyst stage, and then gastrulation occurs. From the gastrulation, they form into a planula larva. So you have to remember this: planula larva, ciliated planula larva. The planula larva, once it is swimming freely in the water, after some time they attach to a substratum, and once they are attached to the substratum, development occurs. They form into scyphistoma. Scyphistoma, with the help of the tentacles, they have those nutrition. They grow to form a young strobila. Strobila divides into buds because of the nutrition that they're getting. And from young strobila, they become an adult strobila. And once they form an adult strobila, the budding occurs. Each bud is removed out from the strobila, and each bud of ephyra is a free swimming one. And after some days after the development, they form into an adult male or a female Aurelia. I hope this is okay with you all. Did you all understand this reproductive system? So whatever the screen that I'm showing, all this matter is what I've told till now. So I'm not going to touch over there. So formation of the planula larva, I told you all, they have the ciliations, the blastophore become ciliated. Yes, Arif?
Ma'am, one individual gives many what do you
Exactly.
say,
One
Aurelia?
individual. Yes, exactly. That is what I was been telling about. One individual planula larva is going to give this many individual Aurelias by budding. Okay. So all these segment, each segment is going to give one ephyra larva, one ephyra, free ephyra, which will form into an adult one. So here when you count, there are 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. A dozen ephyra is present over here. So from one single planula larva, you're going to have a dozen Aurelias. Got that point?
Okay, ma'am. Thank you, ma'am.
Yeah. Welcome. Okay, so this is the planula larva that I talked about, so ciliated larva, and the formation of scyphistoma over here. What all I talked about, I've put it in a matter over there. You can just go through that. So scyphistoma, budding formation occurs, and they form into the ephyra over here. This is an early ephyra and a late ephyra with now can you see the early ephyra and a late ephyra. Early ephyra is having only a mouth, the four-cornered mouth, and as they are developing, they form this radial canals over there. The interradials, the perradials, the adradials are formed, and they ultimately forms into an adult Aurelia. So formation of ephyra also I've also talked about, so I'm not going to go much into the detail over there. You can just go through that. So this is another picture of ephyra larva over there. And yeah, so metamorphosis. I want to talk about a little bit of metamorphosis. As the growth proceeds, the mesoglia increases enormously, and the two layers of the endoderm fuse to form a solid gastrodermal lamellae, except in the regions of the gastrovascular canals. And with the appearance of numerous marginal tentacles and fororal, they form into an adult Aurelia. So here you will find alternation of generation in Aurelia. What is an alteration of generation? That is, in a sexual generation is represented by an adult Aurelia, which is produced by an asexual polyp forms. So that is why it's called alteration of generation. That is by means of scyphistoma larva or scyphistoma, you call them. And this scyphistoma is produced sexually from an adult Aurelia. So alteration of generation. Aurelia is going to give rise to a scyphistoma. Scyphistoma is again going to give rise to Aurelia. So alteration of generation of asexual and sexual reproduction occurs over there. And polymorphism in Cnidaria. Polymorphism means many forms is polymorphism. What is this many forms in polymorphism in Cnidarians or Aurelia? Occurrence of polymorphism guarantees a well-organized division of labor between several individuals. Division of labor. Each tissue forms, each cell forms an organ, tissue forms an organ. Now, why we have different organ system is because you want to have a proper division of labor. So, for example, a driver is going to only drive. You can't expect him to cook. Cooking, you have chefs. Teaching, you have teachers. You can't ask a cook to teach, a teacher to cook, and a cook to drive. So each one has a division of labor. So everyone are experts in one field. So that is what is polymorphism over here. Well organized division of labor between individuals. And the coelenterates are different individuals. They get united to form a colony, and hence polymorphism is a very important feature in this phylum. The best example over here is hydrozoa. And what are the two main forms of polymorphism over here in hydrozoa? They are the polyp form and they are the medusae form. The polyp forms are tubular ones with a mouth surrounded by tentacles, and they have a pedal disc over there. This coral. Yeah. Hello, who's that? Aisha? Yep. Okay, when you have this polyp form, when you're talking about the corals are, they secrete some solid form, and from that solid secretion, they try to grow themselves into a polyp form. The solid substratum that they have secreted in the early life is what is called as corals. We remove that, we shape them and give that bead-like structures. So the solid structures that is present above, where the polyp is growing above that solid structure. This solid structure is the one that they remove from the water. They polish them. They give that shape, different shape, tubular shape, circle shape, different shape, and that is called as a coral that we wear as necklaces or beads. Okay. Medusa is an umbrella shape or a bowl shape tentacles. They have those projections, and they have a concave surface and a convex surface. Generally, polyps are sessile, they don't move, and medusa are motile, they keep moving. But there are some homologies present in them. There are some organs, the tissues that work in the same way, something like tentaculocytes, something like dactylocytes, and gonocytes. They have a homology. Homology is the same type over there present in them. So that is what is. Let me just switch on the battery over there. It's running down. Okay, sorry, I forgot to switch on. Okay, so that's a basic feature of this medusa or the polymorphisms present in them. Coming to the pattern of polymorphism. What is this pattern of polymorphism? The pattern is they can be either dimorphic, tetramorphic, or polymorphic. What is this dimorphic, tetramorphic? If you come to this polymorphisms over here, when you talk about dimorphism, it is made up of two types of colonies over there. So what are they? That is the zooids, two types of zooids. What are they? Two types of zooids. They are only this dactylozoids and the tentaculozoids that are present over here. So they have only two types of zooids, hence the name dimorphic. Dimorphic polymorphism is exhibited by many hydrozoan colonies like Obelia or Turbularia as the example over here. So they have only two types of zooids, dactylozoids and tentaculozoids are present in them. Now, when you're coming to, so this is what I was talking about, the gastrozoids or the dactylozoids and the gonozoids that are present over here. So that is the two types of zooids in the dimorphic characters. Example is the Obelia and Tubularia. Now coming to trimorphic, tri means three types of zooids are present. I told you all in the earlier class also what are the zooids and what are they helpful for. So in trimorphic, tri is three, dactylozoids along with gastrozoids and gonozoids. And dactylozoids, what are they? They are non-feeding defensive structures consisting of batteries of nematocysts. Remember that Portuguese man of war, you have those battery-like nematocysts which helps in stinging. So that is what is dactylozoids are all about. And gastrozoids, what is a gastrozoid? They help in digesting the food, the capturing and taking the food inside. Gonozoids for the production of the reproductive germ cells. Polymorphic. Polymorphic means they have too many number of zooids. What are they? For example, this Hydractinia. They consist of 5 types of polyps. What are they? They have this gastrozoids. The gastrozoids are present over here. They have this dactylozoids for protection, and they have this tentaculozoids, the tentacles or the tentaculozoids for the sensory functions. And they have this skeletozoids, the skeleton or the skeletozoids, which produces the spiny projections of chitin, the spiny projections of chitin that is present over here, and the gonozoids for reproduction. So that is all about the different types of zooids that you find in the polymorphic nature. Now, what not only in the polyps, there are some modifications also in the zooids of the polyps. What are they? The gastrozoids are also called as feeding polyps. They help in feeding polyps, the blue color ones over there. They have a mouth and a long tentacle structures present in there. So they have the gastrozoids with a mouth and a long tentacle-like structure, which helps them. Then they have this dactylozoids. Dactylozoids are a protective polyp over here. So they have a protective polyp. They have no mouth, but have a basal long tentacle, which helps in self-defense. Okay. The gonozoids are reproductive polyps. So they are the reproductive polyps for production of medusa forms over here. So this is a medusa form with a pneumatophores, the nectophores, the orophores, so different types of polyp formation of the medusa. Modifications of medusa. So what are the modifications of the medusa from the polyp form? Nectophore is also called as swimming zooids. They are also called as swimming zooids. So this is a nectophore in the medusa. So this is helpful in the swimming of the zooid. They have muscular bell without tentacles. Pneumatophores, the Portuguese man of war, pneumatophores, they help in floating, bladder-like medusa. Phylozoid, here you can see in this picture, yeah, this is a phylozoid that is present in the Hydractinia. What are they helpful for? They are also called as bract, and they are studded with nematocysts, which helps in protection. And the gonophore or the gonozoids. Importance of polymorphism. Why should these cnidarians have polymorphisms? Because again, division of labor, different function assigned to different individuals rather than only single cell doing all the work. The division of labor would be a better option so that the animal can survive for a longer period of time. You can't make only one person do all the work. The work has to be distributed. Okay, so polyps, they are associated with feeding, protection, reproduction, while medusa forms are concerned mainly only with the reproduction mechanism. So when you need a polyp form, the coral form as such, the coral reef that you find over there is only about the polyps that is present, and they are associated with feeding, protection, and asexual reproduction, whereas medusa is only for reproducing the polyp form. So that is all about the polymorphisms in Aurelia. So we come to the end of our class. And this is the earliest form of the cnidarians that you find is almost 580 million years ago. They have found this particular structure. So that is about the cnidarians. Any doubts? Any doubts? I hope you have.
No, ma'am.
Yeah.
No, ma'am.
Okay, so
You
we come to
explained well.
end. Thank you. So in cnidarians, you will be asked about talk about the obelia structure, or they will also ask you to talk about the Aurelia, type study Aurelia. They might also ask you to talk about the canals, the interradials, the perradials, and the adradial canals. And they will also talk about polymorphism, discuss the polymorphism in Aurelia. So you are going to talk about this
Polyp.
polymorphism. That is polymorphic nature, the diplozoids, the tetrazoids, the trizoids, and the polyzoids. Okay, so you're going to talk about all those zoids, the dactylozoids, the gonozoids, the what are the other one? Nematozoids. So you're going to talk about all those zoids over there. So yeah.
And what about coral reefs, ma'am? Is it important?
Coral reefs, did I give any important points on coral reefs in your textbook? Because I don't have a textbook, so I'm not very sure.
Yes, it is a brief description
Discussion
about. Yeah,
Okay, so you
coral
might
reef,
get a little
barrier
bit.
reef.
Yeah.
Yeah,
You can just go
atoll
through
and
that.
fringing reefs
Yeah, you might
with
be asked
structures.
you might be asked for a short question, barrier reef, right? So you can just go through that.
Okay.
If an examiner is interested, he might.
PDF.
Yeah, this will be shared after every class. I'll definitely share the PDF, so you don't have to remind me. Yesterday I had 4 or 5 students asking me for the PDFs. Instead of individual students asking me each and every time, why don't you form a group and you just ask me in one group? I already sent this to 2 groups. Please make another group so that I can send to all of you all in one format. Okay, so
Okay, ma'am. Thank you.
to keep sending each one of them, it will be difficult for me, right? Okay, then see you all in the next class. When do we have our next class?
We don't know.
Next Saturday. Next
No
Saturday.
idea. Next Saturday is, what is next Saturday? 20. Okay. Okay, whenever. So yeah, whenever we have a class, yeah, you can just call me up. And I think
24th
Sunday we don't have. Do we have on Sunday because it's a Christmas? You people will be celebrating Christmas. Nobody's celebrating Christmas. Okay.
No.
Anyways, I'll check up. You will also be getting an information. So
Yes.
until then, see you all. Okay then. Goodbye.
Bye.
Bye, madam. Thank you.
Bye.
Bye.
PDF of the same will be shared, ma'am.